So this is the third, and I think the last of the prep videos for the lecture 21. And I had finished off, I was introducing the idea of co-transformation. And it's a very quick and a relatively easy experiment that can just give you a sense of whether two genes are close together in the example that I was talking about, within 8,000 base pairs of each other, or whether they're far apart. And so I think where I'd finished was just I'd shown that if you've got genes that are 20,000 bases apart, and you're breaking DNA into pieces that are 8,000 in length on average, then it's impossible for you to find a single piece of DNA that's around this size that's going to include both of the genes. Okay, and so the only way they could possibly get incorporated into a bacterial cell is if the cell takes in one piece of DNA in one transformation event, but then also takes in a second one in a second event. And this is rare. Yeah, and so this is just a physical kind of picture that shows that if the genes are far apart and we've got relatively small bits, there's very little, almost no chance, well, no chance at all that they'll go in together, and it's very rare for each of them to be incorporated in two separate events. You would have to take the probability of this event happening multiplied by the probability of this event happening. So, suppose this was a probability of 15% or something, and this was a probability of about 10%, then you'd expect the probability of both occurring is 0.1 times 0.15 or 0.015, right? So that's a relatively small number of cells that are going to take that up. Okay, but if they're only 1,000 base pairs apart and our pieces of DNA are 8,000, there's going to be lots of pieces of DNA that include both of them, and so they will show up way more frequently together, way more frequently than this rate. This is the probability of them being independently taken up by the bacterial cell, and then we use the actual co-transformation frequency we observe in our experiment to determine, is this approximately the same? So if it was 0.019 or something like that, then we'd say, probably that's about the same. But if it was 0.33 or something like that, that's more than twice this number. So that would be what we would consider a higher transformation frequency. Okay, so basically, if you think about these two transformations being independent events, then that takes us through this. On the 2 slides back, I had given at the end of the last recording, I gave a frequency for, I think ARG was this one, 0.1, and the HIS was this, and so 0.009. So if we looked at those 500 colonies, we would expect to find 4.5 that could grow on that third plate, and the third replica plate was the one that had no arginine in it and no histidine in it. So the only cells that could grow would be wild type for both of those genes. Okay. And so if we do this experiment, though, I've talked us through that with us already kind of knowing the distance between the genes. But if we do an experiment where we don't actually know, which is the reason why we would do the experiment, and we can see a co-transformation frequency that's higher than a number like this, then we can conclude that the genes are within 8,000 base pairs of each other, and that if the frequency is a lot higher, they might even be right next door to each other. And if it's just a little bit higher, then they might be, around 8,000 apart or 7,000 apart. So the higher the frequency is, the closer the two genes are together. So the main message, when the genes are close together, they're transformed together quite often. There will be lots of pieces of DNA in the sample that are carrying both of those genes. If they're far apart, we will see them co-transform together sometimes, because there is that occasional time when bacteria will take up two separate pieces of DNA in two completely separate events. But that is relatively rare, and you would multiply the probabilities of the two individual events to get that frequency. So when we do an experiment, you'd be given data and you'd figure out what the co-transformation or the transformation efficiency of one gene was, what the transformation efficiency of the second gene was, and then what we would expect the co-transformation efficiency to be if they are taken up separately, and then the real co-transformation efficiency to see if it's about the same as what we predicted, if they're independent, or if it's a lot higher than that. So that's the kind of way you would approach that question. Okay, and there I will send Emily some practice questions on that for tutorial. There is no tutorial in the coming week. We canceled it, but there will be tutorial the week after, and that's the week before your midterm. So it's a good, good to practice all those things. So I've talked a little bit about replica plating, and I want to go through how we determine the genotype of each colony, because when we talk about HFR mapping, we're going to be looking at large numbers of cells. So suppose we isolate 1,000 colonies and we have to actually plate them on lots of the same 1,000 colonies on lots of different plates with different selective media, and keep track of each cell which made a colony, and then figuring out what the genotypes are from those results. So it's quite a lot of work, and there's a bit of logic to it. And in the actual lecture, I'm going to present you with some data, and then we're going to work together to figure out what all the genotypes are, just as an exercise, because I think it will really help with understanding. But so here's our experiment. Suppose we have an HFR strain that's wild type for all of these, but sensitive to strep, and an F- strain that's mutant for all these and is resistant to strep. So we use strep resistance so that we can kill off any of these cells after our mating experiment is completed. And we have this marker, which is bio. Bio is the one that enters soonest. And we are going to select for the incorporation of this in our progeny cells. And we are also going to select for threonine. We happen to know from interrupted mating experiments that threonine enters the cells later than the others, but we don't know the exact order and the exact proper distances between them. Okay, and so selecting threonine plus and biotin plus are ways of ensuring that these F- cells have gone through some sort of mating process and that some genes have been incorporated, and then the question will be which ones, right? And we can figure out by the number of crossovers, basically, which we'll talk about in the next lecture. Okay, so we're selecting against the HFR and the F- in our early plating, and then we're going to look at the genotypes of our colonies. Okay. So again, we're selecting for threonine plus and biotin plus and strep resistance. Okay, and so the cells that we collect, we're going to know what their genotypes are at some position. So we're going to know they're all strep resistance because they grew on the plate. They're going to make their own biotin because we didn't provide it to them, and they're going to make their own threonine in there. And so now what we want to know is what are the genotypes for these other genes. Okay, so suppose we only collected 400 cells on this plate and we wound up with 400 colonies. Because arginine and methionine are included in the media, we have 4 different types of genotypes. They're all going to be wild type for threonine, but then some of them are going to be wild type for the other 2 genes, wild type for arginine but not methionine, wild type for methionine but not for arginine, and mutant for both arginine and methionine. So we can replica plate these on different types of media and discover which are which. So how would we distinguish? So this will grow on minimal media, right? Because it's able to make all of its own nutrients. So these cells, they won't grow on minimal media, but they will grow if you add methionine, right? And these cells will grow on minimal media plus arginine. And then these will be the rest of the cells. So the thing to think about, though, here, minimal media plus methionine, these cells will grow too. It doesn't matter to them if there's methionine in the plate, so they'll grow with it or without it. And so therefore, we're going to have to do a bit of arithmetic with the number of colonies that grow on each plate to sort out, okay, how many of them grow on minimal media, and then when we add methionine to it, how many more of the colonies grow that weren't able to before. For minimal media plus arginine, how many grew that were not able to grow on the minimal media, and then the remainder are going to be this genotype. So we will work on this in the class. I think working through it together with everyone, discussing and voting, is going to be the best way to understand that. This is what the replica plating process looks like. So you have your master plate, and of course this is not nearly enough colonies. There's usually a few hundred on them, and you actually have what is called a reference mark. So you have like a piece of tape or something right in a particular position on your block. This is a block that is the same shape as the inside part of your plate, so that when you take the plate and put it on there, it fits right down over. And then there is a autoclaved velvet on here. And when you touch the lid is off, of course. When you touch the plate down and press onto the velvet, some of the cells of the colonies are captured on that, or stuck to it. And then if you put a clean plate down on it with the very same reference mark, then you can look at the colonies that grow on the plates. And so if we're looking at these, and all we want to do is figure out, okay, are these AMP resistant or not? So they're on a non-AMP plate. You can take them and zap them down on there, and then you put the plate aside, and you can grow the colonies up again. The cells will keep growing. But then you take these 2 plates, one that doesn't have ampicillin, one that does, and you plot them down, and you figure out which colonies grew that on this plate, and then you compare to the ones that did not grow on the ampicillin plate. So these guys are AMP resistant, but the ones that didn't grow are AMP sensitive. And so you always have these replicas with this kind of media and that kind of media where we can follow. If you hadn't made a reference mark on here that corresponded to the exact position on the plate, it could be really hard to identify, which of these colonies was which genotype. Because when you go back to the master plate, you might want to grow up a certain number of the AMP sensitive ones to test further for some other purpose, right? So you have to sort of be keeping track of the various colonies and where everything is located and so on. And again, in our case, we would have plates that lacked arginine and methionine, so those would be our minimal media plates, and then minimal media plus arginine, so they're just lacking the methionine, and minimal media plus methionine, they're just lacking the arginine. So that's what I discussed on the previous slide. And then you're going to end up with some numbers. And I would like you to think about figuring out with the numbers that we generate in the class which gene is in the middle and how you can tell. If you have to rewrite them in order, figure out the order, and then think about how many crossovers we're talking about. So we're going to be counting crossovers. And we're going to be working with a linear piece of DNA and a circular bacterial chromosome. We will have selected for, in our experiment, threonine plus, and then, you know, think about the rest of the crossovers. And in a previous lecture, we will have gone through some of the geometry of this and sort of how, you know, you've got to have an even number of crossovers for a circular chromosome, etc. And so that will be coming up in the next lecture.